please stay tuned to the end of this program or see the show notes for important information regarding today's speakers and the content of this podcast. Hello, everyone, and welcome to another episode of Allergy Talk, a roundup of the latest in the field of allergy immunology by the American College of Allergy, Asthma, and Immunology. For today's episode, we will be reviewing 3 more articles from the November-December 2020 issue of Allergy Watch, a bi-monthly publication which provides research summaries to college members from the major journals in allergy and immunology. You can also earn CME credit by listening to this podcast. For information about CME credit or to read archive issues of Allergy Watch, head over to college.acaai.org/publications/allergywatch. And finally, please watch out for continued discussion on this topic on the ACAAI community on DocMatter. We'll have key talk takeaways and engaging question answer with the opportunity for ongoing conversation about today's topic. Hello again. My name is Jerry Lee. I'm an associate professor at Emory University and the co-host of Allergy Talk. And I'm once again joined by my 2 co-hosts. First, Dr. Stan Fineman.
Hello, everybody, and thank you. I'm currently the editor-in-chief of Allergy Watch. I'm in practice here in Atlanta. I'm also on the clinical faculty at Emory.
And then my other co-host, Dr. Maren Curavilla.
Hello, everyone. This is Maren Curavilla, and I'm an assistant professor of allergy and immunology at Emory University.
So we have a potpourri of articles covering a lot of very interesting topics. So I think we should just get right into it. So Stan, I think you have something more to tell us about chronic rhinosinusitis phenotypes and endotypes.
Exactly. In fact, that's this article. In fact, you reviewed this for Allergy Watch from the Journal of Allergy Clinical Immunology. It's entitled "Type 2 Inflammation in Chronic Rhinosinusitis Without Nasal Polyps: Another Relevant Endotype." And this is from some excellent researchers, mostly in Europe. And basically, they start out by pointing out the fact that, in general, phenotypically, the chronic rhinosinusitis patients are classified with nasal polyps and those without nasal polyps. And really, 20% have nasal polyps, 80% just have plain chronic rhinosinusitis without nasal polyps. So we know that. We also know that, in general, the endotyping has shown that the patients who have chronic rhinosinusitis with nasal polyps generally have had the type 2 based eosinophilic inflammation in about 80% of the patients. On the other hand, the chronic rhinosinusitis without nasal polyps are generally considered to be primarily type 1 to type 3 inflammation that's associated with neutrophils and regulated by other cytokines such as IL-6, IL-8, IL-17, and TNF-alpha. So not the type 2 cytokines that we all know and are familiar with. So what these researchers did was a cluster analysis. They basically looked at the 2 different immune responses in patients who had chronic rhinosinusitis without nasal polyps who had had endoscopic sinus surgery. So they were looking for that, and that's what these researchers did. They analyzed mucosal tissue. This is tissue from the surgeries of 240 white patients who had chronic rhinosinusitis without nasal polyps, who had the functional endoscopic sinus surgery, and the endotyping studies that were carried out, and the cases were classified according to various markers, the cytokine expression markers. And the findings were then analyzed with reference to the clinical characteristics of the patients, including the recurrence of the sinus disease, comorbid asthma, the presence of allergies, also blood eosinophilia, and neutrophil counts as well. So the type 2 immune response with the IL-5 concentrations above a certain cutoff of 12.9 picograms per gram of tissue was found in 49% of patients who had the chronic rhinosinusitis without nasal polyps. And this pattern was also associated with increased levels of IL-4, of eosinophilic cationic protein, even of Staph aureus endotoxin-specific IgE. And these patients had this type 2 immune response. They also had increased blood and tissue eosinophils as well as eosinophilic inflammation, such as Charcot-Leyden crystals, some eosinophil extracellular trap death, so to speak, and they had long-term recurrence rate. So the long-term recurrence rate was 19% for patients who had type 2 chronic rhinosinusitis without nasal polyps versus the 9% who had non-type 2. So, obviously it doubled the risk of having a recurrence when you had the type 2 characteristics. So the prevalence of comorbid asthma was 30% in the type 2 and only 9% in the non-type 2. So 3 times more likely to have the asthma comorbidity as well. So about 3% of the patients with the type 2 chronic rhinosinusitis also developed nasal polyps within a 12-year period. So there was evidence, they felt, of the type 2 immune responses in about half of the patients undergoing sinus surgery for chronic rhinosinusitis, even without nasal polyps. And they felt that this shows significant eosinophilic inflammation, higher rates of clinical recurrence, and also rates of asthma. So a specific, phenotype of this cluster analysis of these types of patients. And what you mentioned in your review, Jerry, was that there may be this subset of patients who have chronic rhinosinusitis without nasal polyps who could respond to maybe some of these type 2 targeted biologics. And, that's a suggestion for a different type of treatment. So even if they don't have the nasal polyps and they have the chronic rhinosinusitis, we maybe should consider them candidates, and we could talk about that too.
I mean, we always think about the unified airway again, and certainly these patients, not all of them had asthma, but a fair number of them had asthma, are probably having the same thing top to bottom. I think what was just disappointing is there's really not a good way to identify T2 chronic rhinosinusitis with nasal polyps. Yes, there's, I guess, a general trend and an elevation of these markers, but even the non-type 1 patients, the non-type 2 patients, apologies, had elevated IG and eosinophils. I know it was significantly higher in the type 2 patients without nasal polyposis. I think IgE was actually pretty significantly higher overall, which is a successful biomarker. But still, I think patient selection is going to be very important. I can't imagine a situation where we're just going to throw this on everybody when we don't really have a good justification, unless we get better biomarkers.
I think I had reviewed a paper on AllergyTalk a few months ago, just looking at the efficacy of dupilumab in patients with asthma and concomitant chronic sinusitis that was not stratified by the presence or absence of nasal polyps. That was sort of like a post hoc analysis of the Quest study. And in general, patients with comorbid chronic sinusitis just tended to have more asthma exacerbations and overall higher levels of type 2 biomarkers. And in addition to a more prominent effect on asthma control, dupilumab was also associated with a significant improvement in SNOT-22 scores in patients with concomitant chronic sinusitis. So it would just be interesting to see if something will actually get approved for that chronic sinusitis indication. I know that last year we were looking at possibly, there were trials looking at GB001, which is a prostaglandin D2 blocker specifically for the indication of chronic sinusitis, but then the news was just released that it did not meet any of its primary endpoints in the trial. So it's been sort of abandoned for this purpose.
But I just think patient selection is everything, right? Like,
right.
Clearly not everyone's going to respond. How do you find the people who are the right candidates for this therapy?
I remember the inclusion criteria for that particular trial evaluating the prostaglandin D2 blocker was based on the presence of blood eosinophils. So they did stratify patients with potentially like type 2 inflammation.
So in this study, it's interesting. If you look at the different tables, you could see that even in the non-type 2 chronic rhinosinusitis patients without nasal polyps, their median blood eosinophilia was just under 200, whereas the type 2 patients, their median was about 300. So it was a little higher, but definitely it was still elevated in the non-type 2. So that's a challenge. Now, there was a much greater differentiation in the tissue in the biopsies when they did the after the surgery. They could see a lot of eosinophils in the tissues in the type 2 and practically no eosinophils in the non-type 2. But the blood, they did have eosinophils in the non-type 2.
I think we mentioned earlier about sampling of the upper airway and, running expression, gene expression, that sort of thing. Maybe that's what we should be doing. Or should be considering when we're considering these therapies, right? Getting some sort of biological information through either mRNA and real-time PCR or, again, nasal washes. Speculative, but clearly we need to find the people who respond to these therapies.
Right. And to continue our conversation from the previous episode, maybe doing those gene expression studies would not be a bad idea, even in patients who are started on like biologics, for instance, to see why some are responders and some are not.
Oh, absolutely. Yeah, absolutely.
So one of the things I did want to mention, since you're talking about doing all these analysis and gene therapy, gene analysis, when I started practice, I'm gonna pull the old person card right now. We used to do nasal eosinophils. So we would swab the nose and measure their nasal eosinophils, and that would help us predict whether or not they were allergic, whether they had allergic triggered asthma, and it was part of our evaluation of patients. And obviously, we don't do that anymore, but maybe we're coming back to it.
And we're coming back. We got to sample. We got to go where the problem is and get information. For sure. We just throw medicine at it. We need to understand what's going on. I completely agree with you. The best way to do that's we're not sure. We're not there yet. So, Maren, I think you have the next article, and this is very interesting about a whole another realm of mast cell activation that I did not even know about.
Yes, so this is a really interesting paper, and it was published in the European Respiratory Journal earlier this year and reviewed in Allergy Watch by Bradley Chips. It highlights the heterogeneity of mechanisms underlying asthma, and specifically the role of inflammation and pathways of inflammation in non-atopic asthma. So we know that mast cells in the lung can directly promote contraction of airway smooth muscle and bronchoconstriction by releasing leukotrienes and histamines through activation of the high affinity IgE receptor. But mast cells are also involved in allergen-independent processes capable of causing bronchoconstriction, for instance, in exercise-induced asthma, in which bronchoconstriction is believed to be caused by hyperosmolarity and thermal changes of the airway lining associated with exposure to cool and dry air. So therefore, IgE-independent mechanisms of mast cell activation also exist and can mediate bronchoconstriction. And one mechanism of airway smooth muscle contraction involves this ubiquitous calcium-permeable cation channel. It's called TRPV4, which stands for transient receptor potential vanilloid 4. And this TRPV4 is a channel that is activated by a whole range of stimuli: osmotic stress, low pH, mechanical stimuli, heat. It's been implicated in several different pulmonary processes like cough, asthma, COPD. Like I said, it's widely expressed, but it's not present on mast cells. It's expressed on airway smooth muscle. And TRPV4 stimulation has been shown to increase airway smooth muscle, intracellular calcium, and bronchoconstriction. But it's been previously demonstrated that this TRPV4 mediated airway constriction seems to be entirely dependent on leukotrienes, and it can be blocked by montelukast. So trying to make some sense of why TRPV4 stimulation on airway smooth muscle causes leukotriene-dependent bronchoconstriction, the authors of this paper performed this series of experiments that were just very creative to delineate the mechanism by which TRPV4 activation causes bronchoconstriction. So first, they showed that the TRPV4 agonist, GSK, long series of numbers, not important, showed a pro-contractile effect on airway smooth muscle, and they showed this using tracheal tissue preparations that they isolated from both guinea pigs as well as human lungs. And these effects were blocked by pretreatment with a TRPV4 antagonist. But also, the application of montelukast as well as xylutone inhibited TRPV4 stimulated contraction of airway smooth muscle, confirming the role of leukotrienes in this process. And then they looked at cultured human airway smooth muscle cells, wherein the TRPV4 agonist that they used increased intracellular calcium. And to try and link the capacity of this calcium influx and airway smooth muscle contraction, they used this imaging technique that I've never heard of called tachography, which sort of looks at three-dimensional characteristics of the cell and links the measure of cell contraction with changes in intracellular calcium. So the point of going to that was that surprisingly, tachography showed that TRPV4 agonist-induced increase in intracellular calcium actually did not cause cell contraction in these cultured airway smooth muscles, suggesting that the activation of TRPV4 on airway smooth muscles causes the release of a mediator that activates mast cells that releases leukotrienes, causing bronchoconstriction. And then they did other experiments showing that TRPV4 stimulation causes ATP release from smooth muscle cells, and this stimulates mast cells causing bronchoconstriction, and the effect can be blocked by leukotriene inhibitors. And finally, because protease activated receptor 2 or PAR2 has been known to couple with TRPV4 and is also expressed on every smooth muscle in humans, the authors looked at the ability of the endogenous PAR2 agonist trypsin to try and reproduce the effect of this TRPV4 agonism, and trypsin-stimulated contraction of human airway smooth muscle was again blocked by montelukast. So the conclusions were that TRPV4 activates mast cells and thereby causes airway smooth muscle contraction. These TRPV4 channels on the airway smooth muscle can be gated either directly using this synthetic agonist that was used in these experiments or through activation of the protease activated receptor 2 on airway smooth muscle. And TRPV4 activation results in ATP secretion, mast cell receptor activation, mediator release, that is leukotrienes causing bronchoconstriction. And I thought it was interesting that the protease activated receptors coupled with TRPV4, because we do know that this plays a role in mold triggered asthma or fungal or severe fungal asthma. And so what the authors postulated is that the same axis that starts with PAR2 and then signals through TRPV4, ATP, mast cells, ultimately leukotriene release, could play a role in bronchoconstriction that is associated with fungal asthma. So essentially, this TRPV4 dependent allergen independent mechanism could act concomitantly with allergen dependent activation of mast cells and augment their effects. And interestingly, they also provided a mechanism for the GERD asthma relationship. They mentioned that TRPV4 is activated by low pH, so reflux may exacerbate asthma by triggering TRPV4 and thereby indirectly causing mast cell activation and bronchoconstriction. So I thought this paper was very exciting because it just really very nicely elucidated sort of additional mechanisms of non-allergic asthma and may even herald or illustrate new therapeutic agents in the future.
So that's very interesting. Did they comment on inflammation, meaning potentially, I would assume cysteine leukotriene production, mast cell activation would be more of an eosinophilic phenotype, but I don't want to speak out of turn because you mentioned these other mechanisms. Do they talk about the inflammatory profile?
No, they did not. They did
Hmm.
not. They just sort of honed in on those leukotrienes, which made me like sort of reconsider my whole approach of never giving anyone singular.
So, we know that there are people out there who respond to montelukast. It's just that we don't know who these people are. You wonder, maybe those montelukast responders, meaning montelukast better than inhaled steroid. That's really what I'm suggesting. Maybe they are something along this mechanism. Completely speculative, but clearly that montelukast is the better drug for them for a reason. And maybe this is one of them.
Yeah, I thought it was interesting. There was a editorial along, paired with this, and they were pointing out that this TRPV4, mechanism is responsible for a number of things. It's been implicated in things like cough, COPD, lung edema, and even exercise-induced asthma. And the question is, are we going to start developing therapeutic agents that target this?
That would be great. We need another weapon. That would be fantastic. The more the better, for sure. So that's just such an interesting article. Thank you for sharing that one, Maren. So I'm going to round out the trio of articles. And I don't think we've talked about a immunology article in a while. I'm sorry, immunodeficiency article in a while. That's really what I meant. So I got to mix it up. So for people who are real fans of immunology, clinical immunology, this was a very interesting article, just sort of a heads up from the group up at Mass General Hospital in Boston. And so what they did is they are examining IgG and IgG subclasses, but examining sex and racial differences. And again, I've never really considered that before. Certainly, I consider age. I have all these referrals where the patient's flagged to be hypogam, and then you just apply the normal range that is appropriate for the age, and it goes away. Easiest consult to do. But not really much known about other groups and whether the normal ranges should apply to those populations. So, what they did in this article is they looked at their entire patient data registry. So there's this Partners Research Patient Data Registry. It spanned between January 1st, 1989 and March 15th, 2018. And they queried all the patients who had an IgG subclass performed. Now, just to put the caveat here, that's all comers. We're going to understand people getting IgG subclasses, there's probably something wrong that people are looking for. So by no means am I stating that this is a healthy cohort, but to do exploratory research, you got to start somewhere. I think it's quite reasonable to start in this fashion. So, with that caveat, they look at patients who are adults. So these were 18 above. So we're not worried about the pediatric population. And basically, they pulled out of the chart normal demographics like, date of birth and race, address, and diagnoses, because they wanted to get certain categories. And you can imagine what those are: immune deficiencies, autoimmune diseases, infection, pulmonary diseases. They were trying to collect some information. So when they looked at their population, we could start with gender. In general, the most significant difference that's related to gender between the IgG and the IgG subclasses is IgG4. So interestingly, IgG4 is generally higher in males than females. It was 56.3 on average milligrams per deciliter for males versus 37 for females. And certainly we use IgG4 potentially for diagnosis of IgG4 related disease. So there's this idea that maybe we should consider what would be our cutoff for an elevated IgG4 if we're thinking about that. Maybe. Now, the race information was really interesting to me. Full disclosure, I'm East Asian. So looking at total IgG, the highest mean IgG level was Black patients, 1,504. Asians next, 1,340. White, 988. So 988 white, Black, 1,504. That's 50% higher on average. Now, again, I'm talking about broad averages here. Now, let's talk about the subclasses. Subclasses, Black patients, 938. They had the highest IgG1. White patients, 592. Asians, somewhere in the middle. IgG2, Asians have the highest IgG2 levels of 493 compared to whites, 305. Blacks, again, in between those two. IgG3, Black patients have the highest IgG3 levels, 91.9 for Black patients, 55.9 for white patients, Asians in the middle. And then IgG4, very significant. Asian patients, 140 milligrams per deciliter was their average IgG4 level versus Black and white patients. Black, 53, white, 41. Again, we're talking almost triple the amount of the concentration. So again, very exploratory research. They looked at the effect of patients undergoing immunodeficiency workup. Obviously there might be some patients undergoing some sort of immunodeficiency workup, maybe hypogam. They eliminated those patients, still had the same observations. And clearly, I don't know the diagnosis of every single patient here, so it has to be validated in a healthy cohort. But interestingly, and I didn't know this, me being East Asian, it's possible that I have a higher IgG4 level at baseline than other populations, and potentially that would affect how I'm evaluated, how people think of me when they're considering differential diagnoses, and other racial groups might be identified by different ways. For example, if you have a very high baseline IgG, hypogam, your definition potentially should be different if you're a Black patient, which you have a higher baseline IgG level. Again, clearly I'd be more interested in a healthy population, but these gender, race differences and how we apply normal ranges seem to extend beyond more than age. And again, we should just be aware of it. Clearly, we work better. I mean, we treat the patient, we don't treat numbers, but we do use numbers to support our diagnosis. So it's always a good lesson just to think about potentially what other factors in place when we're interpreting results. And of course, we do functional testing. That's the other thing, right? We don't.
So, Jerry, let me just jump in here. It's not that, with the way you were analyzing it, we do know that the IgG1 is the largest component of total IgG, and IgG4 is the smallest component in terms of the total percentage. So I'm not sure, what these numbers mean.
I think the implication of the authors was particularly about the role of IgG4, how that's used in clinical practice as the diagnosis of IgG4-related disease. But you're right. I mean, in clinical practice, at least I'm not sure of a specific cutoff where a patient would potentially be misdiagnosed. But again, that would require further investigation as well.
Yeah, I have no comment. I can't remember the last time I ordered IgG subclasses. So.
Honestly, that's the only reason I order it. I only order it if I was worried about IgG4 related disease. You see some patient with really bad lymphadenopathy, and you want to figure what's going on there. I mean, I guess technically, if you had like an autoimmune pancreatitis patient or something, obviously a lot of those patients don't see us, by the way. But something.
Although, IgG4, we're using that in research to look for immune response for our immunotherapy. I mean, that is clearly correlated. I mean, the higher IgG4 is specific. It's a specific antibody, but that's what we see responding to our immunotherapy, whether it's oral immunotherapy or subcutaneous. It's an IgG4. So I agree with Baron. I don't order subclasses, but I think the role for our specific IgG4 type measurements are for measuring immune response.
Yeah, and we're learning so much about T2 responses and the role of IgG4. I think on a previous, I'm not sure if it's this podcast or a journal club, there's correlation with very high IgG4 and eosinophilic esophagitis, food specific IgG4 and eosinophilic esophagitis. So, as we learn more about these conditions, the actual interpretation of the results are more helpful. Clearly, by no means am I implying that is the only role for that test. But, in terms of measuring the total IgG subclasses, I know some people think about immunodeficiency by measuring subclasses, but generally the proof's in the pudding. It's going to be functional testing that's going to back that up as well. So anyways, I thought this was a very interesting issue of Allergy Watch. Please go to the college website and read the rest of the fine articles that the other assistant editors and Stan reviewed. And please rate our podcast if you enjoy what you're listening to. We welcome your feedback, corrections, and suggestions. The email again is allergytalk1word@acaai.org. And I hope everyone have a wonderful 2021. It's been a heck of a year, everybody. All right. Have the wonderful rest of the day. The ACAAI is presenting this podcast for educational purposes only. It is not medical advice or intended to replace the judgment of a licensed physician. The college is not responsible for any claims related to the procedures, professionals, products, or methods discussed in the podcast, and it does not approve or endorse any products, professionals, services, or methods that might be referenced. Today's speakers have the following disclosures. Dr. Lee has nothing to disclose. Dr. Curavilla has been a speaker and consultant for GlaxoSmithKline, and Dr. Fineman has been a speaker for Takeda and has done research with Aimmune, DBV, and Biocryst.